Okay, so continuing our lecture, we were talking about an important biochemical organism that is present in our body and is continuously working so as to fulfill our biological needs. Yeah, so we are talking about enzymes. So we saw about the chemistry, we also saw about its nomenclature, and now we were going to see about the classification. And last will be the mechanism of action and the factors that affect an enzyme activity. Yes, kaya factors ઉપર enzyme work કરે છે, kaya factors એને affect કરશે, say for example pH છે, temperature છે, and also the substrate. So depending on these three things, there will be affection. So we already saw about the chemistry. They are formed from proteins. Yes, they are the tertiary and quaternary structures with globular proteins. We have seen the classification of proteins, and accordingly, they are these types of proteins. They have important active site, which can also be a binding site or a catalytic site. Binding site એટલે કે substrate join થશે અને product બનાવશે, and catalytic site એટલે કે બે substrate join થઈને એક outcome product આપશે. Yeah. Cofactors, these are non-proteinous things that that join with the enzyme to form an active site. Yes, cofactor એટલે કે એવી વસ્તુ or એવા ions or molecules જે protein નથી પણ attach થશે enzymes ને અને active site બનાવશે, that is active proteins બનાવશે અને પાછું process માં help કરશે. Inorganic cofactors and organic, we saw both the things. In inorganic, we saw zinc, magnesium. So these are the ions. Organic can also be there. So glucose phosphorylase is a small organic molecule. Pyridyl phosphate. So phosphorus is a glycogen. Phosphorylase is necessary. Okay. Moving to substrate. So substrate is something that will attach to an enzyme. The enzyme સાથે attach થઈને એક product આપશે. Yes, so જે attach થશે, that is substrate. So reactant in biochemical reaction is termed as substrate. When a substrate binds to an enzyme, it forms an enzyme substrate complex. So it will form a complex. After this complex is formed, there will be formation of product depending on if there is enough amount of substrate or not. So enzymes ક્યાં બનશે, આપણે વાત કરી કે this is the function. This is how it will attach, but where are they formed? So they are synthesized in ribosomes of rough endoplasmic reticulum. Rough endoplasmic reticulum. Endoplasmic reticulum again are of two types, smooth and rough. Rough શું કામ હોય, એની ઉપર ribosomes હોય. Ribosomes માં proteins બને, or you can say enzymes synthesize થાય. Yes, information ક્યાંથી આવે ribosomes ને બનાવવાની? તો from the nucleus, from the DNA. Intracellular extracellular enzymes, so જે enzymes એક સેલ માં બન્યા એમાં જ એ કંઈક ફંક્શન કરશે, એમાં જ કોઈ પ્રોસેસ ને મેટાબોલાઇઝ કરશે, that is intracellular. Each enzyme જો બહાર જઈને કંઈક કરશે, so that is example of extracellular enzyme. Example of intracellular is oxidoreductase catalysis biological oxidation. Yes, it is a type of enzyme. We are going to see the same in classification. આજે જે આપણે classification કરશું એમાં oxidoreductase is one of the types of enzymes. Yeah. And enzymes involved in reduction of mitochondria. So each cell માં produce થયું છે અને ATP માંથી ADP બનાવવા માટે, ADP માંથી ATP convert કરવા માટે જે enzyme use થશે each same cell માં, that is example of intracellular enzyme. Extracellular enzyme, that is secreted somewhere but its function is somewhere else. Yeah, so the example that we saw for digestive enzyme was pancreas. Yes, it was secreting a few enzymes which are not helping pancreas anything, but they will help in digestion when they are moved to other parts of the digestive system. Characteristics are most important. Yes, characteristics of enzyme, along with you can say some other one or two things. What is enzyme or which are the types of enzyme can be asked again as a 5 or even 10 marker question. Yes, so keep this in mind. Enzymes speed up the reaction by lowering the activation energy of the reaction. So enzymes ekdam speed કરી દેશે reaction ની. Speed of the reaction means A plus B gives C in 1 hour. Yes, so if in presence of enzyme, that A plus B will give C in 10 minutes. Yes, so that is the difference an enzyme will make. Their presence does not affect the nature and properties of the product. Yes, see substrate ne change કરશે, substrate સાથે attach થશે તો જ substrate enzyme complex બનશે, but it will not ever change the product, that is properties of the product. જે product બનશે એની કોઈ properties change નહીં કરે, never. If it does, it is not an enzyme. Yeah. They are highly specific in their action. That is, each enzyme can catalyze only one kind of substrate. So depending on which type of substrate will attach to them, they are very specific. Yes, एक enzyme एक substrate સાથે attach કરીને એક product જ બનાવી શકે. It is predetermined. Yes. Small amount of enzyme can accelerate chemical reaction. Yes, એવું નથી કે substrate 100 છે તો 100 જ enzyme હોવા જોઈએ. Even small amount of enzymes are there compared to substrate, and also they can formulate or fasten the process. Enzymes are sensitive to change in pH, temperature, and substrate concentration, as we already saw, and we are going to see a bit detail about pH માં કઈ રીતે change થાય છે, ક્યાં સુધી એ allow કરી શકે temperature માં, and substrate કેટલું હોય, તો એની ઉપર enzymes activity will vary. Turnover number is nothing but defined number of substrate molecules transformed per minute by one enzyme molecule. So જ્યારે પણ કોઈ એક enzyme substrate સાથે join થશે તો એ કેટલા substrate ને per unit of time per minute change કરી શકે, any ability, that is termed as turnover number. So turnover number naturally is 6 into 10 raised to 6 per minute. Yes, so 6 into 6 followed by 6 zeros. Yes, per minute આટલા substrate ને change કરી શકે per minute. We are talking per minute. Nomenclature, as we already know, ASE પાછળ લગાડી દેવાનો, no matter what the process is, and depending on that there will be the name. So maltose plus water, maltase. Yes, that will be the enzyme. Lactose plus if it is degradation of lactose, it will be lactase. Yes, and similarly there will be examples that we already saw, but there also were some enzymes which are named before this nomenclature. આજે system આવી એની પહેલા ઘણા બધા enzymes nomenclature wise આપેલા હતા. Yes, so pepsin, trypsin, renin, these are examples of all the three are of course digestive enzymes, but they were named before this system came in, so it is predetermined. એટલે ઘણા enzymes એવા પણ હશે જેનું નામ AS થી ASA થી and nahi thatu hoy, but still they are considered enzymes. Why? Because before the system came they were already named. એનું નામ પહેલેથી આપી દીધું હતું. Lactose gives product to glucose, and galactose enzyme will be lactase, and similarly this already we have seen. Yeah. Moving to the classification. Now classification is for 6 major classes. Enzymes 6 types na hoy em કહી શકાય. Yeah. Systemic international classification is there. So International Enzyme Commission has given this classification. Classification is based on the reaction catalyzed. આ 6 કોના ઉપર આપ્યા કે કયા reaction માં enzyme help કરે છે. Depending on that there are 6 types. Each class further divided into subclasses, subclasses, and so on. Yes, એટલે એક example એ oxidoreductase enzyme છે તો એના અંદર પણ બીજા classes હશે, એના પણ classes હશે, but we are not bothered to go in that much detail. Yes, we are not going to study that science in detail, but we should know at least the 6 major groups. Yeah. So these are the 6 major types of enzyme. They are all dependent on the reaction type. So first, oxidoreductase help in reduction and oxidation, redox reaction. Yes, something from chemistry 11th 12th. So example, lactate dehydrogenase. Yes, so as the name suggests, it will be, the substrate will be lactose. Yes, and it will be dehydrogenase, that is reduction reaction will be there. Yeah. Transferase, so it moves chemical group. So say for example COOH group, carbonic acid group can be moved to CHOH group, aldehyde group. Yes, so hexokinase, so transferases or transferase type of enzyme, these are responsible for moving chemical groups. એક chemical group માંથી બીજા chemical group માં convert કરે. Yeah, so that is transferase, as the name suggests. નામ જ છે transfer કરું. શું transfer કરું? chemical group. Yeah. Third, hydrolase, that is hydrolysis, bond cleavage with transfer of functional group of water. Hydrolysis, water ne break down કરવું or to form water in reaction, that is hydrolysis. Yes, નામ ઉપરથી જ ખબર પડી જાય, hydrolase, remove the water molecule. Yes, so it will decrease or remove the bond and form a functional group of water. Lysozyme, yeah, that is an example, lysozyme. Lysase, non-hydraulic bond cleavage, so it will break all non-hydraulic bonds. Yes, example is fumarase. Isomerase, as the name suggests, intramolecular group transfer, isomerization. Yes, we know what is isomerization. Mirror image. Yes, enantiomers, all these things that we saw in the proteins. Yeah, so that is in the carbohydrates and proteins both. So enantiomers and isomerization, that is the same process we had described. triose phosphate isomerase, and last ligase, that is synthesis of new covalent bond between subtypes or substrates using ATP hydrolysis. So ATP breakdown thase and નવું covalent bond બનાવશે, that is the function of ligases. Example, RNA polymerase. Yeah, it is simple. Oxidoreductase reduction oxygenation reaction કરાવશે. Transferase chemical group transfer કરશે. Hydrolase, it will be responsible for formation of water molecule by breaking of bonds. Lysase, that is non-hydraulic bond cleavage. Isomerase, formation of new isomers. Ligase, synthesis of new covalent bond between substrate. Yeah, simple things. You need to remember the reaction type and one example of each. Yes, whenever asked for classification. So also classification of enzymes can be asked for 5 marks or more. Yes, classification itself with addition of anything else. Yes, what is enzyme? You can also name few enzymes. What are they formed of? Their normal characteristic functions and classification. Together they can ask for 10 marks also. Yeah. Coming to mechanism. બધું જ જોઈ લીધું આપણે કે સ્ટ્રક્ચર શું છે, નામ કઈ રીતે આપશું, ક્લાસિફિકેશન શું છે, ફંક્શન. Now we see the mechanism. If they are joining, how are they joining and how they will form a new product from the same substrate. Yeah. So there are 4 main actions: covalent catalysis, acid basis catalysis, catalysis by proximity, and catalysis by strain. All the 4 we'll see in detail, but first remember covalent catalysis, covalent bond related. Second, acid base catalysis depending on the pH. Catalysis by proximity, just coming near to the substrate, and catalysis by strain, that is under pressure. Yeah. So first, covalent catalysis. Enzymes from covalent linkages with substrate forming transient enzyme substrate complex with low activation energy. Enzyme is released unaltered after completion of reaction. So અહીંયા શું થશે? Substrate is coming, it will form a bond, covalent bond. Covalent bond એટલે કે electrons share કરીને જે bond બને, that is covalent bond. Yeah, so it will share electrons and according to that it will form a covalent connection with the enzyme substrate. આવીને covalent bond બનાવશે enzyme સાથે, connect થશે અને એમાંથી product બનશે. Yes, after the product is formed, enzyme એવું ને એવું બીજા substrate સાથે bind કરી શકશે. Why? Because there is a covalent bond that is formed. Covalent bond break થઈ જશે, substrate product માં convert થઈ જશે. Yes, simple thing. So here substrate is coming to form bond here. Yes, A is one substrate, B and C are already together another substrate. Yes, they are changing the shape. Enzyme no shape change થાય છે. Once they have left, say for example A and B bond થઈ ગયા અને C એમને બહાર નીકળી ગયું. Yeah, so again A, B and C are same. You can see A, B and C from the first and the third figure, they are same. So now pachu A molecule eklu and B, C molecule together, if they come and they can bind together, and again this enzyme can be used to form AB and C. Yeah, same example, this is covalent catalysis. Covalent bond change karu, that is covalent catalysis. Second, acid base catalysis. So mostly undertaken by oxidoreductase enzyme, as the name suggests, oxidation reduction reaction through acid base. Catalysis is mainly done by oxidoreductase enzyme. Mostly at the active site, histidine is present, which can act both as the proton and proton acceptor and donor. So koi pan vastu jo proton donate kare to is acid, proton accept kare to is base. Yes, we have seen the acid base in the pH first few lectures. Yes, so the same thing will be following here. So histidine, we have taken the example. So histidine can do both things. They can either be a donor, proton donor, or they can be even the proton acceptor. H plus દઈ પણ શકે, OH minus accept પણ કરી શકે. Yes, both things are possible. So they will change depending on the acidity and basicity of the catalyst. Yeah, substrate જે છે acid છે, base છે, એના ઉપર ઈ catalysis કરશે અને નવું product બનાવી આપશે. Yeah, catalysis by proximity, just depending on the distance. એટલે કે નજીક આવે તરત substrate product માં convert થઈ શકે. Yeah, in this catalysis molecule must come in bond forming distance. Yes, એક bond form થાય એટલા distance ઉપર આવી જાય. Say for example A and B, they are coming in contact with here. ઈ contact ના જ લીધે એક molecule બની ગયું, that is your product. Yes, ઈ contact ના જ લીધે ખાલી molecule બની જાય, that is product. That is catalysis by proximity, just coming near the distance. So when enzyme binds, a region of high substrate concentration is produced at active site. તો અહીંયા જુઓ substrate, બે substrate છે, તો અહીંયા high concentration substrate થઈ ગયા, બે substrate થઈ ગયા. This will orient substrate molecules spatially in a position ideal for them. So this is an ideal position to form connection between the two substrate, and this will form a new product. આ બંનેને form કરવું છે. Yeah, this is like two individuals who want to buy something. Yes, who want to buy the same thing. So what they will do, they have to go on, say for example, just a second. Okay, so sorry, there was a call. Yeah. So in the catalysis molecule, catalysis by proximity, so they will come in contact. They just need a place where they can bind together. Yes, so they will bind together on the substrate. So on the enzyme. So enzyme પાસે બે positions છે. They are coming here. Yes, and because they are in proximity, નજીક આવી ગયા છે, for that reason they'll form the product. Yes, simple as that. So this will orient the substrate molecule spatially in a position ideal for them to form a product. And by bond strain, as the name suggests, it will be bond strain. Strain means pressure. Pressure આપીને શું થાય? It will break the bond. So you can see this is the substrate. It will come here because there is enzyme is able to break the bond. It will break the bond and two new products will be formed. Yeah. So mostly by lysase. Lysase means breakdown. Yes, કાઈ પણ breakdown કરવું હોય ત્યારે bond strain use થાય. Enzyme will act as a bond strain. Enzyme substrate binding causes reorientation of the structure of site due to strain condition. Now અહીંયા જુઓ, પહેલા first it will bind here. Yes, depending on that there will be change in the structure of the enzyme. The enzyme changes shape slightly as the substrate binds. Once this change in the shape occurs because of the change in the shape of the enzyme, the substrate breaks into two parts and there will be two new products formed. Thus transitional state is required, and here bond is unstable and eventually broken. In this way bond between substrate is broken and converted into products. Yes, so bond જ્યારે break કરવું હોય ત્યારે mainly use થશે catalysis by bond strain. Yes, so these are the four important mechanisms of enzymes. We'll revise once. This is important. Yes, this is the only way enzymes work. આ four ways માંથી એક way માં enzyme કોઈ પણ જગ્યાએ કામ કરતું હશે. Yeah. So first is covalent catalysis. So કોઈ એક substrate નું covalent bond break કરીને બીજા covalent bond આપવું. Yes, so that is covalent catalysis. Second, acid-base catalysis. So taking example of histidine, एक proton donor and एक proton acceptor. Both can be only one thing, and depending on that જે સામે substrate હશે એની સામે એ proton આપશે or accept કરશે. Yes, so it can change the property of that individual substrate. Catalysis by proximity. Two substrates just need to come together at a specific position. Enzyme position આપશે અને નવું product form થઈ જશે. And last is the lyase, that is catalysis by bond strain. So it will break the bond. Catalysis પાસે આવશે, enzyme પાસે આવશે substrate, shape change થશે, shape change કરશે enzyme, એના લીધે bond break થશે, એના લીધે there will be no two products. Yeah, so these are the four main types of catalysis or mechanisms of enzyme action. Now, how does this happen? We initially told you, I had told you about a lock and key mechanism. Yes, so substrate no shape એવું જ હોવું જોઈએ કે જે enzyme ને match કરે, opposite હોય exact. Yes, like a lock and a key. એક lock એક key થી જ ખુલશે unless and until there is a duplicate or something. Yeah, so એક lock એના opposite shape વાળા જ key થી ખુલશે. Yes, so that is the main lock and key model that was given by Emil Fischer in 1894. Okay. એન્ઝાઇમ છે એ લોક એન્ડ કી મેકેનિઝમ ઉપર વર્ક કરે છે અને પ્રોડક્ટ ફોર્મ કરાવે છે. સો લોક એન્ડ કી હાઇપોથેસિસ assumes the active site of an enzyme are rigid in its shape. Yes, so say for example આ સબસ્ટ્રેટ છે તો આ એન્ઝાઇમ નું સ્ટ્રક્ચર છે તો તે ઇટ સેડ લોક એન્ડ કી મોડેલ શું કહે છે કે ફિક્સ છે કે શેપ આવો જ હોય એન્ઝાઇમ નો બહુ જ ફિક્સ છે અને એની સામે સબસ્ટ્રેટ એવો આવશે તો જ એન્ઝાઇમ એક્ટિવેટ થશે અને કામ કરશે યસ otherwise it cannot it is a fixed thing rigid there is no change in the active site before and after a chemical reaction એટલે એમનું એવું કેવું છે કે ઈ ચેન્જ ના થઈ શકે એક્ટિવ સાઇટ જેનો શેપ છે ઓલરેડી એન્ઝાઇમ જે રીતે બન્યું છે ઈ એવું જ રહેશે લાઈફ લોંગ જ્યાં સુધી ઈ ડિસ્ટ્રોય ના થઈ જાય ડીનેચર ના થઈ જાય યસ that was lock and key model then came induced fit model કે જે પ્રમાણે સબસ્ટ્રેટ આવશે એ પ્રમાણે એન્ઝાઇમ અડેપ્ટ થશે yes that is induced fit model this is proved now most recent studies have revealed that the process is much more likely to involve an induced fit model what is induced fit model so according to this exposure of an enzyme to a substrate causes a change in the enzyme which causes the active site to change its shape to allow enzyme and substrate to bind એટલે કે સ્ટાર્ટિંગ માં જે વસ્તુ હતી કે આમાં કીધું તો લોક એન્ડ કી મેકેનિઝમ માં કે સેમ યસ આ ફિક્સ હશે યસ કે સબસ્ટ્રેટ એક જ આવીને અહીંયા જોઈન થશે ઈ જ પ્રમાણે રહેશે એગ્રી પણ જ્યારે આ સબસ્ટ્રેટ પ્રોડક્ટમાં કન્વર્ટ થઈને નીકળી જશે that will change the shape you can see here yes this is કે સબસ્ટ્રેટ પહેલા છે that is agreed yeah then sorry enzyme પહેલા છે એનું shape fix છે active site fix છે in a shape substrate is coming attaching substrate lyase no example છે yes it is breaking the bonds it is moving forward now here there is slight change in the shape of that enzyme there will be slight change yes a slight change નવા substrate ને પણ અને નવા enzymes ને પણ activate કરી શકશે yeah so that is induced fit model એટલે કે shape of an enzyme is fixed when it is formed પહેલી વાર જ્યારે કોઈ સાથે substrate સાથે attach થશે enzyme ત્યારે fixed છે agreed yes but later on after ઘણા બધા substrate જો attach થશે અને product બનતા જશે તો એનો પણ shape change થતો enzyme નો પણ shape change થતો જશે yes that is induced fit model induced because of something it is changing yes and it is fitting new things so induced fit model yes so first the substrate will be this shape later on the substrate this substrate will be able to join along with this other substrate can also join of different mechanism of different shape why because the active site keeps on changing yes slightly with every new substrate attaching એક એક નવું સબસ્ટ્રેટ અટેચ થશે નવું પ્રોડક્ટ ફોર્મ થશે એના લીધે એક્ટિવ સાઇટ થોડીક થોડીક ચેન્જ થતી જશે and so it is induced to fit other substrate and enzymes also now coming to enzyme kinetics that is the last part so how do enzymes are affected કઈ રીતે affect થાય કોનાથી affect થાય પહેલા તો what is enzyme kinematics so branch of biochemistry in which we study the rate of enzyme catalyzed reaction yes rate at what speed rate means rate of change or rate of moving so that is the speed of an reaction so it કેના ઉપર depend કરે અને કેટલું હોય yes all these things are studied under enzyme catalyzed reaction kinetic analysis reveals the number and order of an individual step by which enzyme transferred substrate into product એટલે આ branch એમ કે through this study we will know કે number and order of individual steps કેટલા steps લાગે છે as well as number of reactions that are done to transform a substrate into product a plus b gives c c to d d to e તો a plus b આપણો substrate હતા e is the main product તો એની વચ્ચે બીજા ત્રણ steps આવ્યા yes and e પણ કેટલી સ્પીડ માં ચાલે છે yes a plus b to c is fast c to d is slow d to e is again fast yes so that all rate is also determined under the study of kinetic analysis studying an enzyme's kinetics in this way can reveal the catalytic metabolism of that enzyme its role in metabolism and activity is controlled or how a drug or an agonist might inhibit the enzyme now આ બધું સ્ટડી કરવાની જરૂર શું આપણે કે રિએક્શન ફાસ્ટ છે સ્લો છે કેટલા રિએક્શન્સ થાય છે તો પ્રોડક્ટ બને છે આ બધાનું સ્ટડી કરવાનું એક જ વે કે આપણે આપણા મેટાબોલિઝમની ખબર પડે યસ ઓલ એન્ઝાઇમ્સ આર નેસેસરી ફોર મેટાબોલિઝમ ક્યાંય પણ આપણે બધા રિએક્શન્સ આફ્ટર ધીસ વી આર ગોઈંગ ટુ સ્ટાર્ટ સે ફોર એક્ઝામ્પલ કાર્બોહાઈડ્રેટ તો ગ્લાયકોલિસિસમાં પણ એક એક રિએક્શન જે છે there are 12 main steps so in each step there will be enzymes તો એન્ઝાઇમ્સ ના હોય તો શું થશે આપણા બોડીમાં યસ ઓર આપણે એ પ્રોસેસ હજી રેપિડ કરાવી હોય તો we want to improve the glycolysis by drugs બહારથી એવા drugs લેવા છે બહારથી એવા enzymes અંદર induce કરવા છે કે process fast થાય or slow થાય yes so it is important to study the kinematics of a enzyme that is why it is utmost important
Enzyme catalysis, it is increase in rate of reaction with the help of enzyme. Yes, as we know, catalysis is nothing but helping something. Like a teacher is a catalysis for a student. Yes, if I am not teaching, if no one is teaching you biochemistry, you will learn it probably in 2 or 3 years, but we are trying to do it in 6 months. Yes, so I am, or a teacher is trying to do it for much more faster for you. Yes, so that is an catalysis. It is an increase in the rate of reaction with the help of an enzyme. Catalysis by enzyme that proceed via unique reaction mechanism typically occurs when the transition state intermediates forms a covalent bond with the enzyme. That is covalent catalysis. We know that we have seen the example also before. During the process of catalysis, enzymes always emerge unchanged at the completion of the reaction. Yes, enzyme ma koi divas koi change avse nahi. Yes, its nature, its physical, chemical properties will remain same even before and after the reaction. તો જ enzyme કહેવાય. એ પાછું use થઈ શકે તો જ એને enzyme કહેવાય. If it is not being used again, that means it is not an enzyme, it was part of a substrate. Which are the factors that affect the enzyme catalyzed? We talked about 3 of them: temperature, pH, and substrate concentration. Yes, so we'll describe again this. So first is temperature. So temperature માં કઈ રીતે affect થશે? So raising the temperature increases the rate of enzyme catalyzed reaction by increasing kinetic energy of reacting molecule. Yes, so kinetic energy is directly proportional to increase in temperature. કોઈ પણ વસ્તુ increase in temperature થશે તો enzymes ની activity વધશે, but there has a limit. Up to 40 degree, 50 degree it can increase. પછી શું થશે? પછી એના સ્ટ્રક્ચર ને ડેમેજ કરવા માંડશે. એના સ્ટ્રક્ચર ને ડેમેજ કરશે એટલે એન્ઝાઇમ ડીનેચર થવા માટે, ડીનેચર થઈ જાય એટલે પછી ફંક્શન નહીં કરી શકે. Yeah. Enzyme work maximum over a particular temperature known as optimum temperature. Yes, enzymes are very specific to temperature. જે enzymes એક specific temperature માં work કરે એ temperature ને કહેશું આપણે optimum temperature. Enzymes for humans generally exhibit stability temperature up to 35 to 45 degrees. Yes, so for normal our body we need 35 to 45 degree temperature only. એનથી જો વધારે હશે તો enzyme કામ નહીં કરી શકે, destroy થઈ જશે. એનથી ઓછું હશે તો પણ કામ નહીં કરી શકે. Temperature coefficient is a factor by which the rate of biological process increases for 10 degrees Celsius increase in the temperature. So every 10 degree biological process કેટલી વધે છે? Yes, say for example 35 compared to 25 degree, 35 degree. Yes, so that will increase for normal human being. For biological processes it is 2 times. It is 2 times. That is, jo 5 substrate convert thata tha 10 ma. Yes, 5 substrates were being able to convert into product, you can say. So 5 substrate 10 માં convert થાય 25 degree. Double થઈ જશે at increase in 10 degree. Yes, this will be normal up to 45 degree. પછી 45 degree થી વધશે તો enzyme નહીં કરી શકે કાંઈ. It has to die off. It will die because of the temperature. However, sometimes heat energy can also increase the kinetic energy to a point that exceeds the energy barrier, which results in denaturation of enzyme. એટલે કે enzyme destroy થઈ જાય. More than 45 degree, jo 50 degree poche, you can see from this only. Yes, so this is the graph. So below 5 degree, the enzyme will be inactive. Yes, 0 or minus degrees of body temperature. We are talking about body temperature, not outside temperature. We are talking about body temperature. So here it will not be active. 5 degrees ma on be, your blood and everything will stop. You will die eventually when your body temperature is so low. Yeah. So enzymes will also die. It from 5 degree to 40 degree, it is increase in activity. Increase in activity depending on this temperature coefficient. Yes, 5 થી compare karo 15 ne, compare karo 25 ne, compare karo 35, compare to 45. Yes, so it will be double. Yes, so અહીંયા say for example rate of reaction was only 2 substrates were being done for, અહીંયા 4 થતા જશે. 2 ની જગ્યાએ 4, 4 ની જગ્યાએ 8, 8 ની જગ્યાએ 16. Yes, so it keeps on doubling for a normal biological process. But when it reaches 40 or 45 degree and later you try to increase the temperature, it denatures and it will die off. Any activity ochi thati jase. And up to 60 it will be completely off. After 60 there is no enzyme that is active. Your body will lose all enzymes if your body temperature rises more than 50 or 55 degree Celsius. Yeah, so this was the effect of temperature. Coming to second, pH. Yes, so again pH is also optimal. Yes, after blood pH બહુ જ વધારે buffer system, the whole buffer system that we described, that is specifically for to maintain pH. pH શું કામ maintain કરવું પડે? enzyme activity કરી શકે. enzyme બધી activity કરી શકશે તો જ તમારું normal metabolism ચાલુ રહેશે. Otherwise there will be diseases in the body. So effect of pH, rate of almost all enzyme catalyzed reaction depends on pH. Most enzymes exhibit optimal activity of pH value between 5 and 9. Yes, so 5 and 9 ની વચ્ચે જ આ pH ની વચ્ચે જ બધી normal activities થશે. High or low pH value then optimum value can cause ionization of the enzyme, which result in again denaturation of the enzyme. So again, say for example an enzyme in its structure has a capability કે 6 to 7 pH માં work કરી શકે, તો એનથી ઓછું or એનથી વધારે તમે એને ટર્ન કરાવો તો શું થશે? It will die because it will be denaturated. Denaturation means protein નો જે structure છે એ change થવા માંડશે. protein નો structure change થઈ જાય, enzyme નો structure change થઈ જાય, that means it is of no use. જેની માટે બન્યું તો એ નહીં કરી શકે. Yeah, so you can see again from the graph, from increasing 5 up to the 10 it is okay, but maximum is at around 7 and 8. In between 7 and 8 our normal body and blood pH is to be maintained at 7.35 to 7.45. remember this. Yes, normal body or blood pH is to be maintained at 7.35 to 7.45. Now pH also affects the formation of hydrogen bond and sulfur bridges in proteins and so affects the shape. Now pepsin, trypsin and arginase, we have taken 3 examples. Depending on these 3 examples, you can see some enzymes, they work in acidic condition. As you know, in our stomach the pH is very low because of secretion of HCl, hydroxychloride acid. Yeah, so because of this there will be increased amount of H plus ions, and so the pH will decrease. pH is inverse log of H plus ions. Yeah, so pepsin molecule, this has optimum, you can see from 0 or 1 it has optimally up to 5 only it can work. આપણે હમણાં જોયું 5 થી 9 normally enzymes work કરે છે, but this is a special case. So pepsin, it works from around 0 pH to up to 5 pH. 5 pH થી વધારે હશે તો એક્ટ જ નહીં કરી શકે. Yes, it will die because it is formed as such for the digestion purpose. Pepsin secreted in the stomach, so for the digestion of it, it works only in this pH. Opposite 5 to 9 વાળો example લઈએ તો trypsin. Yeah, it starts main activity from say around 5 and it will reach its maximum at around 7 8 and will die off more than that. Yeah, and at times there are other pH specific like arginase, which is also secreted in the small intestine. Yeah, so there the pH is opposite, basic. So here also it starts its action from 5 and can move at peak at 9 pH. Yeah, and then pachi die off thwa mande. So it is not ke 5 to 9 ni vacche જ રહીએ. There are variations. The variation depends on the need of the body. Yeah, so stomach હોય તો stomach માં pH બહુ ઓછું હોવાનું, તો એમાં pepsin ને કામ કરવું હોય તો 5 to 9 નું ના ચાલે. So its structure as such, proteins એમાં એવા હશે કે જે withstand કરી શકે. The structure will be as such which can withstand the change in the pH, low pH. Yeah, and high pH માટે પણ we saw the example of arginase. So all the 3 are possible. It can be acidic specific, it can be normal 5 to 9, or it can be also basic. Yeah, so pH has its own effects. Okay, fine, we'll keep it till here. We'll not move forward to Michaelis-Menten model. It will take a bit time. Yeah, okay. Any doubts till now in enzyme? Yes, koi pan doubt enzymes ma till now? We have seen the definition, we have seen the main action, what is the structure of an enzyme, then we saw about the nomenclature. Today we saw about the chemical properties, classification also we saw, and the models, both the models. Yes, induce fit and lock and key. Now it is believed it is induce fit only. If in exam it is asked, you are about to write for induce fit model, it should always be about the enzymes. You should mention both lock and key and induced fit model, and induced fit you should write it is accepted now for all the enzymes. And we saw the effect of temperature and pH on the enzymes. The last part is left. Probably we'll be able to cover it in the next lecture and even start with some of the other topic. Okay, any doubts till now? Anyone, please reply in the chat. Everyone should reply. Okay, fine. So in next lecture, we are going to complete the enzymes and may even be starting with the carbohydrates or vitamins, whichever is feasible. Yeah. Till enzymes, it will be asked in exams for CE. Yes, what all things we have covered will be asked, including enzymes. Completely when we are in the next lecture, we'll be completing the enzymes. So the whole enzymes will be asked along with the pH and buffers, introduction to all the three: carbohydrates, lipids, that is fats, and proteins with enzymes. Yeah, all these things can be asked in exams. Okay, fine. So we'll keep it till here. Ayush, have you taken the attendance?
Yes, sir.
Okay, fine. So we'll keep it till here. Uh, we'll meet again tomorrow and start with the exercise therapy. Move forward for exercise therapy.